Our next speaker is Dr. Jeroen Pollet. He is the assistant professor at Baylor College of Medicine in Houston, Texas, at the section of tropical medicine, and is the director at Texas Children's Center for Vaccine Development. Originally from Belgium, he acquired his PhD from the University of Leuven, which is in Belgium, before coming to the US as a Fulbright Fellow in 2012. He joined Baylor in 2013 as a postdoc and was appointed faculty in 2016. Dr. Pollet has more than 10 years of experience developing vaccines for various parasitic and viral diseases. His research is focused on the optimization of novel vaccine formulations with the aim of improving immunogenicity and efficacy. His group has worked on vaccine candidates against hookworm, schistosomiasis, and Chagas disease to bring them from discovery into clinical trials. Alongside Dr. Peter Hotz, Dr. Pollet's team helped to develop Corbavax, which is a low-cost SARS-CoV-2 vaccine that has been produced by Biological E in India. The vaccine has been approved for emergency use and is currently administered to protect children older than 12. So I'd like to bring over Dr. Pollet.
I'll see if this microphone working? Can you hear me loud enough? Yes. Okay, great. Thank you for the kind introduction, and thank you for the invitation to speak here to a live audience. That's really nice to do that again. So I'm a vaccine developer at Baylor College of Medicine, and I'd like to talk a little bit about how we use the Luminex, among other technologies, to look at cellular immune responses against our vaccines. But before I get into data, I'd like to talk a little bit about the unique structure of our group, because I think it's relevant on how we look at new technologies. So I'm part of the Division of Tropical Medicine at Baylor College of Medicine, and our core mission is looking at neglected tropical diseases. We do surveillance all over the Americas, we develop new diagnostics, and we're heavily looking at the development of new vaccines. We have about 45 people in our labs, and we have 12 faculty members, and our senior leaders are Dr. Peter Hotz and Dr. Melina Bothazi. Both are nominated for Nobel Prize this year, so I'm humbled to be in their shadows. Aside of research, we do education. We have the National School of Tropical Medicine, where we educate the new generation of healthcare people about tropical medicine. We also have people affiliated with the hospitals, so we have hospital at Ben Taub, and also some of our physicians at Texas Children's Hospital. And we also do a public policy. We are heavily fighting against the anti-vax and anti-science movements, and we also try to talk to the government about the importance about these vaccines, because we all know we live in a world where there is migration, and there is climate change, so these neglected diseases are moving up across our borders into Texas. But what's truly unique about us is our science. We have really a unique system, I think, in our group. So all of us are part of Baylor College of Medicine, which is, of course, an academic institute. And there I am a classic PI. I have some students, and they all go for their PhD, or they have other goals. But it's all basic research. It's about innovation, high risk, about publishing papers. But next to that, we are affiliated with Texas Children's Hospital, where we have the Vaccine Development Center, and that's really something you can consider a little bit of a small biotech. And in that, all the good ideas for the academic, we actually move into a product development. And so there we have a larger team, so we have all our PIs come together, and we are really developing a product to bring products from the bench into the clinic. And so there, of course, there is no more room for error. We are really dedicated in making sure that whatever we produce there has a future, so it must be low cost, easy to produce, and we make sure that from the start of our research, we document everything really well. So we really have something what we call an integrated product development strategy, where each PI has their own specialty, and we really connect everything from the discovery phase all the way into the clinic. I'm the leader of the formulation development team, but we have antigen discovery, formulation development, making the product, do preclinical. We do not have a cGMP manufacturing site, but we always work with either partners in the US, typically the Walter Reed Institute, the Army Institute, or what we actually prefer is to work with local institutes in the endemic countries, because if it's produced in that country, it is easier to distribute. We have our own quality control system, so while we work with cGMP manufacturers, we like to look at the control ourselves, the quality ourselves, so the stability of the product. And so we have a whole team that helps us to regulate, to the regulators, to bring really the product into the clinic. We cannot do that, of course, without a wide range of collaborators. We have a wide range of academic collaborators, funding collaborators. We have consulting and support by different companies that sometimes give us materials for free. And of course, we have a strong connection with manufacturing sites all over the world. Midvax, that's in Israel, Biopharma is Indonesia, Biological E is India, Q Biologicals is Belgium, ESA is Japan, so it's really worldwide production sites that we're connected to. And our vaccine development portfolio holds about 14 different kinds of vaccines. As you can see here, there are two platforms. There's a recombinant protein platform, that's kind of our basis. That's also something you can easily develop in low and mid-income countries. But we also started in 2007 with our own mRNA platform. Here you can see the list of all the targets, diseases. So we do a lot of parasites like schistosomiasis, leishmania, Chagas disease, and also a lot of viral infections. In blue, those are things that are in preclinical development. In orange, those are things that are in the clinic, phase 1 or 2. And in green is our only product, which is currently on the market, which is our COVID-19 vaccine. As Bobby mentioned, Corbevax was developed in our labs. Together with the Indian collaborator Biological E, we manufactured this in India. We've made over 300 million doses. And we're currently vaccinating children between 12 and 14 years old, because that group hasn't been vaccinated yet. We also, since the beginning of this week, are good to go. We got a green light in Botswana, so we'll send 100 million doses to Botswana to vaccinate in that region. Of course, developing vaccines, our goal is to induce an immune response, and there's of course two parts. There's a humoral immunity, and then there's a cellular immunity. And most licensed vaccines against infectious diseases, they all provide mostly humoral immunity. It's all about antibodies. And one of the reasons why that is because the cellular response is very complicated. There's a lot of different kind of T cells. They generate all these cytokines, and we do not always know which direction we want to go to. And that is really where the difficulty lies for us, because when we are developing a vaccine, we know we want to have neutralizing antibodies. It's a clear answer. But if we are developing a vaccine, which of these cytokines do we want? Mostly when it's like a new disease like COVID-19, we do not know which cytokines are important. And so it's important that we have new technologies so we can rapidly figure out what is beneficial to clear infections. So the methods that we're using at Baylor are intracellular cytokine staining. So we use the flow cytometry, of course. The advantages there is multi-parameter cytokine analysis, and we can do phenotyping. The drawback is it's very labor intensive, it's costly. You have to fix cells, so if you have very precious samples, you lose that sample. And it's a very complex data analysis, meaning not everybody in my lab can do flow cytometry. The gold standard in vaccine development is ELISpot. You will see if you look at clinical data where people are restimulating PBMCs from patients, it's all about ELISpot. It's kind of the older technology, and vaccine development is kind of conservative. So the advantage is it is as gold standards. It has a high throughput, but of course the big limitation is you can only look at one and maximum 2 different parameters. We are using in the lab, certainly at the development stage, a lot of the Luminex, so the multiplex cytokine analysis. It's very easy to use, it's standardized, that's why we love it. It's very easy to have somebody new in the lab, train them, and they can generate a lot of data for us. The only drawback is that we cannot do phenotyping. Of course, if we would select certain T cells, then we know which T cell it is. But a lot of times we actually look for cytokines in the blood, or we re-stimulate splenocytes, and then you really do not know where the cytokines are coming from. Kind of the future for us, so we're looking at, is the transcriptomics. Specifically, if you work on a single cell transcriptomics, you can have a lot of data from small samples. So in the next, I want to just discuss 2 examples of vaccines that we have developed that I think is interesting and relevant. First, I want to talk about the COVID-19 vaccine, because that's the only one which we did really from the bench all the way into the clinic and the market. And it's also interesting during that development how we looked at cellular data. And secondly, I will talk about our Chagas vaccine, because that's against a parasite, an intracellular parasite. Antibodies have absolutely no working function there, so we really are forced to look at cellular immune response. And the data I will show is interesting because we combine actually mRNA technology and protein-based technology in heterologous vaccination, which gives a very unique cellular immune profile. So let me start with the development of our Corbivax. So the first data, of course, we did in preclinical was a mouse model. At that time, in the original phase, there wasn't a mouse model to infect, so it's all immunogenicity data. And so we vaccinated mice twice, 21 days apart, and we have a vaccine, it's a protein, the RBD, plus aluminum and CpG. And so when we showed our data, for review, the reviewers really only looked at the antibody data. While we did, we used Luminix to look at the cytokine profile, no questions asked. Also, the regulators, it's like, oh, it's a Th1, Th2 balanced immune response, but nobody really goes into depth because we do not know which one is important. Is it important to have interferon gamma? Probably with a viral infection, but all the others, nobody knows what's good and what's bad. So everything is focused on cellular immune response. Then we did studies in non-human primates. Here, this data shows some in-depth analysis of 2 formulations. We did one with the protein and aluminum, and the other one we added a TLR7/8 antagonist in there, so an immunostimulant. And for this, because it's a limited studies, we only have so much non-human primates, we really decided to use flow cytometry to go in depth and do the phenotyping. What you can see here in the data in the red dots is that our adjuvant really stimulates the T cell response. And what was really nice in non-human primates is that we could do challenge tests, and we can really see now, what is important, which parameter can be used as a correlate of protection. And so in the y-axis here, that is after infection, we look at the total RNA copy numbers, and in the x-axis, you see all of the parameters that we're investigating. And here on the bottom left, you see ID50 FRNT, that's the neutralizing antibodies. And you can see if there is a large amount of neutralizing antibodies, we found back a low number of RNA. So there's a clear correlation. For all of these other antibodies, you can see there's a clear correlation between high amount of antibodies means lower number of viruses. But here on the far right, that's the cellular data, and it's kind of ambiguous. We don't know what to make from it, but it's definitely not a clear correlation between what we saw on the cellular response versus effectivity of the vaccine. This is some data from our phase 3 study. Again, in our phase 3, we focus on this data is comparing actually Corbivax against Covishield, and a lot of the focus is on the humoral data. We see significant difference between the 2 vaccines in the humoral data. We did also cellular analysis. We used ELISpot, but we didn't see a significant difference between those 2. And so you could say that really the cellular response was not a driver to look at what is the optimal formulation for a vaccine. But that doesn't mean that we say that cellular immune responses aren't important, because we know they are. And others also know that this is just some data from Moderna. They also focus heavily on antibodies, and they make their decisions on neutralizing antibodies. They present the data from the cellular response, but they don't really optimize the formulations to let them go in a certain direction. If it's a balanced Th2, Th1, that's all you see in the papers. But recent data really highlights the importance of, is that me? So recent data really highlights the importance of the cellular immune response, because as we all know, as we come into the Omicron phase, the vaccines really didn't protect us from getting, infected, but we still didn't get severely ill. And so why is that? We believe a large portion is that because your T cell responses are still there, and they prevent you to get severely ill. And that's interesting because now a lot of data shows the importance of the cellular response, and it's just a little bit more robust against all these variants that are coming up, because single point mutations, they can abolish certain individual T cells, but within each O, it's a single, because they're not focused like in the RBD. These point mutations in the cellular, in the T cell epitopes, they're all over, and so it's not focused in the RBD region. I don't think I push on buttons.
Neutralize that virus.
Yeah. Sabotage.
Can you answer a question?
Yeah, of course.
So when you do the immunophenotyping for the CD4s and CD8s, like how well do you characterize them? Because would it maybe be possible that because you don't see a correlation with the virus going down necessarily with the T cells going up, that you don't look at the specific CD8s or CD4s that would be responsible for that drop?
Yes, and we actually did look in depth, but we never found, for that particular data set, we didn't find a very good correlation. But we look at, for us important is, polyfunctional T cells, but, what are they exactly doing? But we never found a really good correlation in that, in our non-human primates data set. And that's a little bit of the shame, of course. It's the big question that we had is that if we, now see the importance of the cellular data, it's too far, right? Vaccine development is really product development, so we're on track. Nobody's going back to look at, we're going to change now the vaccine formulation to get a different immune response. The ship has sailed. There's no way back. So it's really important that we get data as fast as possible, and antibody data is really easy. It's in ELISA. Everybody can do it, and you have it. Getting cellular data, it becomes more and more easy to do these assays, but interpreting the data is really hard, and connecting the dots.
Hi, this has been super interesting so far, and this might be a naive question, but I wanted to know, how did you use the Luminex to help inform the results that you were getting to analyze the cellular versus humoral response?
So how we exactly did the Luminex experiments? Yeah, so what we classically do is, so we harvest the spleens from the mice, and now we re-stimulate them with either the antigen that we use to vaccinate, or you can of course use a peptide library if you know exactly what the T cell epitopes are. And that's a little bit of a problem in the field, is that there is no standardized methods in vaccine development. Everybody does what they believe is best. And so, if you re-stimulate splenocytes, one of the questions is how much antigen you use to re-stimulate. When are you looking at it? We look at 48 hours, but others look at different time points. And so really it's cellular data. If you look into the literature and we're comparing the different coronavirus vaccines, right? We're always lining them on efficacy or on neutralizing antibodies. Nobody's comparing them on cellular immune response, because the data that we have, you can just not compare them.
Okay, sorry. And so from that, you're looking at the cytokines that were being produced by these cells. And so you were saying it's difficult for you to compare the humoral versus cellular responses because, I know there's a question here because I'm trying to see the connection. Hold on, let me think about this a little bit and workshop it.
Yeah.
But this is
So one of the things, for example, was early on people would say IL-17 really bad, for inflammation and all of that. And then, when you see IL-17, you're like, is that real? I mean, you don't know, but how it goes in literature. If you can find for every cytokine probably someone that says it's really important and someone that says it's actually really bad if you have that. And it's really difficult to navigate into that. Okay, and so here what you see is that's just a broadness of these T cell responses over different, so this is ELISpot over different variants. We didn't see as much difference. If you look at the neutralizing antibodies, they are dropping significantly, but the T cell responses do not. Okay. That's kind of the conclusive remarks here. At the development stage, our data, our knowledge about the cellular immune response was just not fast enough getting to us to really have an impact on how we would develop this vaccine. Everything was based on, antibody responses because that was just the simpler questions. Everybody knows neutralizing antibodies, they block the virus from entry, and everybody's comparing based on that. We don't look at specific cytokine levels, what didn't mean that we didn't continue to investigate the cellular response, and then after the fact, have the knowledge about it. The second example I want to give is the T-cell immune response against a heterologous vaccination, which is first priming with an mRNA vaccine and then boosting with a protein vaccine. Because mRNA vaccines, they give a specific cellular profile. Protein vaccines give a certain profile, but combining them gives actually a third profile. And that's pretty interesting. You need to know, again, first what Chagas disease is. So Chagas disease, it's a disease which is caused by Trypanosoma cruzi. It's an intracellular parasite which has a very complicated life cycle. If you want, you can really start in the Triatoma bug. In this bug, you have these parasites living, and the bug is a block-sucking insect. It will bite you at night, and when it feeds, it will also defecate, and in the feces you have the parasite, and when you are scratching, you're basically scratching the parasite in the bite wound, and that's how you get infected. And then it has a complicated life cycle also in the human body. You have this one with the flagella. It will go into the cells, drops the flagella, and then multiplicate in this form, which is an amastigot. And they all have different surface antigens, and it's very, very complicated. And as you may imagine, an antibody response against such a parasite really doesn't do much. Some antibodies can slow down the flagellar form and some less infection, but it's all about T cells. And we have set up an infectious model in mice, and we've studied for years because it's a chronic disease. So what happens is it's an acute phase first, you overcome it, and then slowly the parasites will move in certain body parts, primarily in the heart tissue. It will cause scar tissue, and suddenly somebody dies of cardiac arrest, and you can see they have a large amount of scar tissue in the heart. And so antibodies doesn't do much. What we know from our models, if we do large mouse studies and we see which ones are surviving and clearing the parasite, is all about polyfunctional T cells, CD8 T cells. And so we have this idea of priming with the mRNA and then boosting with the protein, because we believe that will give us the better polyfunctional T cells. And so we look at, again, we use Illuminex to look at the cytokines, and we have seen there that here in black and in brown, those are the heterologous vaccinations. Orange is mRNA alone, and the purple is protein alone. And it was immediately from the Illuminex data, we could see that the two heterologous ones are really standing out. And so the big question, of course, is it CD8 or CD4 cells? So we looked at the flow, and we see it's a little bit in both, an improvement. And some of them are statistically different. But what really was the best data is actually the polyfunctional T cells here, interferon gamma and IL-2. That's where we really see the difference. And just from other studies that we have done, we haven't done a challenge study with this particular vaccine combination, These challenge studies on chronic diseases take a long time for mice. We actually do year-long studies, but we know from those other studies which profile we want, and we know that this looks very promising. And the big question always is how to get all of your data together. You know, a lot of our PhD students, they drown in the data when they look at cellular immune responses, and I encourage them like to put everything together in this kind of a heat map where we have the complete picture. And this yeah, clearly it's overload. See, the humoral response, what is very interesting is that our protein vaccine is really the best in the humoral response, but we're not really interested in that. This is the splenocyte data, so that's the Luminex data. And here you can see the mRNA protein boost coming up too. But what we're really interested here is the polyfunctional T cells, and there, you know, the prime with the mRNA and the boost of the protein is really the clear winner. And so that's really something we're really interested in pursuing further. Previously, like 3 years ago, this was unheard of. No one would say, well, this is all really nice in theory, but practical. Now you have to make an mRNA vaccine and a protein vaccine, but actually that's not so really hard to do. So we believe there is really a future into looking these heterologous vaccines. So conclusive remarks on that is, you know, we really saw here a very good polyfunctional T cell response. And so we believe that this heterologous vaccination is really the way forward. Now, what is really actually interesting on this is that, so we're talking to manufacturers in Mexico because Chagas disease is endemic in Mexico, to produce our GMP protein and to do actually clinical trials. And so we've shown all of the importance of the cellular data, but the vaccine producer, so one of the parameters of vaccine development is you have to show stability over time and potency of the vaccine. And you would think we're going to show potency with the T cell responses, but that's actually something they do not want. It's way too complicated, and so they're hammering down that they want to have a potency which is based on antibody response, because they say, well, there's a correlation between if there is antibodies produced, there is some kind of an immune response, and thus that means that the vaccine is still potent. So it's a really practical point of view, because that's always, you know, the limitation is that we have, you know, we have flow cytometry, we have Luminex, but if we work together with Mexican manufacturers, they do not always have all this instrumentation, and if they do, then they don't always have the same type of protocols, or they don't have the kits available. So that's always some of the limitations. So some final thoughts and directions from this talk. So the T-cell immunity is really complex. It's maybe too complex for us vaccine developers, because we really usually are working on a really short time frame, and we don't always have the time to look and figure out which cellular immunity is needed. But now different assays and techniques are available, so there's really actually no excuse to at minimum look at all of these parameters. But when you have a lot of data, like in our heterologous vaccination for Chagas disease, it's really, you know, we need to think about how are we going to present this data, because you know, when you make a vaccine, in the end, the final decisions where this can go into humans is the regulators, and then it's the, you know, government policymakers, and you cannot show complicated cellular immunity to them. So it's always a matter of how can we, you know, bundle all of that data into simple understanding. I think if you say polyfunctional T cells, that's something they can understand, say that these T cells attack the bad cells. But of course, if you come with a whole panel of cytokine data, even the regulators, they don't understand which one is the most important. And what happens when, you know, if you would look at the stability of the protein, the potency, and one of the cytokines goes down a little bit, how much is, you know, critical? It's really hard. We haven't found yet where to draw the line. With that, I want to acknowledge all of these people, because of course it's all about teamwork, and we have a really fun international team from all around the world working on these vaccines. If any questions, I'm happy to answer them.
Thank you, Dr. Polet. Any questions for Dr. Polet?
Thank you. Thank you. It was very interesting. I am just curious to know regarding the heterologous approach for the vaccine against Chagas. Is it that the RNA and the protein were against the same target, or are you guys trying to use, different RNAs and proteins together to target different proteins or antigens at the same time?
Yes, so they are the same target. Obviously, if they would not be the same target, you couldn't, So it's really a prime boost approach, the same target. We use a TC24, which is a flagellar protein.
I have a question, actually. With the future directions for Corbevax, what are the plans for it to target against the BA2 variant? We know that right now it's starting to attack more of the stomach and I think the intestines. While I think you're collecting nasal swabs and possibly serum, is there any preliminary comments that you want to say regarding against that variant and what the immune response is against it?
Yeah, so for 2 years we have chasing behind variants. I mean, we have updated all our vaccines, and in the end, it turns out that it's a very complicated story. So the vaccines are currently not being updated, and people are looking, instead of specific variant vaccines, we're looking at the Panzer Bico vaccines that have a broader protection, and it has actually very good reason. If you look at the newest data on Moderna, where they showed actually their Omicron vaccine, where they used it in rhesus macaques, it shows that it is actually not really a good result from the boosting with a specific, and it has to do with original antigenic sin. When you're vaccinating someone with a certain antigen, then it's really hard to overcome the prime vaccination or prime infection, if you want to, because the body recognizes and thinks it knows what it is, and it makes an automated immune response against the original, and even boosting with a variant, it doesn't really work that well. So the future is probably to have a vaccine that covers most of the variants and gives a broad protection. It may not be 100% efficient to prevent infection, but I think most important is that we don't get severely ill and that we can live with the disease around us. But I think it's a utopia to think that we can ever get rid of this particular coronaviruses. We can only hope to get it under control.
Okay, thank you. Any other questions for Dr. Polet?